Hi everyone, my name is Anika and I work as a postdoc in Vlatka Zolda's group for epigenetics at the Faculty of Science, University of Zagreb. And my research focus is the regulation of IgG glycosylation, and that is what I'm going to talk about today. So, we know that IgG glycans can fine-tune the immune response and they are very important in immunity in general, and it's not a surprise that they change in many pathological and physiological states, so in different diseases, all in all when homeostasis is disturbed. But the mechanisms that regulate IgG glycosylation are almost completely unknown. So we know that IgG glycosylation happens in the endoplasmic reticulum and the Golgi apparatus, with the help of different glycosyl transferases and glycosidases, but we don't know how are those proteins regulated and what other proteins are implicated in the process of IgG glycosylation, and to what proportion do they impact IgG glycosylation, and are they specific for IgG, or are there some shared mechanisms between different glycoproteins. And in the last few years, the largest progress was made using genome-wide association studies. So here you can see different genes that associate with IgG glycosylation, and they are found by GWAS. And you can see that there are glycosyl transferases here, like ST6-GAL1 and FUT8, but most of the genetic loci found have no apparent role in IgG glycosylation. Here is a functional network that was created from all of these loci that were associated with IgG glycosylation through GWAS, and there are different types of genes here. So these rectangular ones are actually implicated in the process of protein glycosylation. These green ones are transcription regulators, and these purple-edged ones are implicated in the immune processes. But we currently don't know how exactly do they impact IgG glycosylation, and we decided to try to unravel this functional network using an in vitro approach and using CRISPR technology. So we know that CRISPR is made out of a Cas9 protein and a single guide RNA, and Cas9 protein induces a double-stranded break in a DNA sequence, and a single guide RNA can be designed to be complementary to any sequence of interest. And when Cas9 is targeted via guide RNA, it will induce a double-stranded break. And this approach is used for gene editing, but shortly after the discovery of CRISPR-Cas9, dCas9 protein was created, and dCas9 is catalytically inactive Cas9, and the fun thing is that it can be fused with different effector domains, and when you put guide RNAs into the mix, those guide RNAs will target that effector domain to any sequence of your interest. And this technology, of course, exploded for different types of approaches, from gene regulation, epigenome editing, chromatin imaging, and many others. And it's often used for targeted gene activation or gene silencing. So we can use different types of effector domains, ranging from direct transcriptional regulators like dCas9-VPR for gene activation. For gene silencing, we can use dCas9-CRAB for direct silencing, or some precise epigenome editors, like, for an example, DNMT3A, which is a DNA metal transferase that will actually methylate genes and silence their expression in that way. So what we decided to do is to use these VPR and CRAB effector domains fused to dCas9 protein to construct a transient expression system with stably integrated CRISPR-dCas9 fusions for regulation of genes involved in IgG glycosylation. So this is an in vitro system, and it's designed to study the mechanisms that regulate the process of IgG glycosylation. And VPR is, when it's targeted around the transcription start site of a gene, it will activate gene expression, and CRAB does quite the opposite. So CRAB actually can recruit different epigenome editors that will lead to chromatin condensation and consequently gene silencing. And all of our vectors, our CRISPR vectors, are based on our modular CRISPR-dCas9 molecular toolbox that was developed in our lab a few years ago. And this toolbox enables a one-step generation of an expression cassette with easily exchangeable modules. So we can very easily design any type of an expression cassette with different types of promoters, different types of effector domains, Cas9 orthologs, fluorescent markers. We can use from 1 to 6 guide RNA molecules, depending if we want to target one gene or maybe up to 6 genes simultaneously. But for the functional studies of these GWAS network that are involved in IgG glycosylation, we actually upgraded this system. This is a dual model, so for stable genomic integration and for transient transfection. So this expression cassette that expresses DCAS9 fusion proteins is actually stably integrated into the genome via transposon technology, and this expression plasmid actually codes for IgG and has guide RNAs, and that is transiently transfected later. So just let me give you the workflow of how that works. So the in vitro model are freestyle cells. Freestyle cells they grow in suspension, and they are very easy to manipulate, which is very important because these CRISPR cassettes are very large and very hard to get into the cells, and they are also optimized to secrete large quantities of IgG, which is important for successful glycan analysis. So we first, using our modular toolbox, we assemble our expression cassettes that are then stably integrated into the genome of freestyle cells using piggyback transposon technology. And then we obtain monoclonal cell lines that express either DCAS9 VPR for targeted upregulation or DCAS9 CRAB for targeted downregulation. And then we actually transfect these stable cell lines transiently with an IgG expression vector that also has single guide RNAs for our candidate genes. And this configuration must be emphasized because it obviates any need for selection, because IgG secretion will happen only in cells where targeted manipulation took place. And to validate our system that it works for the studies of IgG glycosylation, we first decided to target the 4 main glycosyl transferases that we know are implicated in IgG glycosylation. So here is the glycan profile in freestyle cells. You can see that majority of glycans are core fucosylated, so we decided to downregulate FUT8. And there is not much sialic acid or bisecting, so we decided to upregulate MGA3 and ST6-GAL1, and galactose is present in about 40% of the structures, so we both up and down regulated before GALT1. And this was just a proof of concept experiment, and these are the results. So when we targetedly manipulated each of these GTs, we actually observed expected changes. For an example, when we upregulated MGA3, bisecting increased on the glycans from IgG that is secreted from freestyle cells. Of course, some non-related structures also change, but that did not come as a surprise because we know that the glycosylation pathways are interconnected on several levels. So, for an example, after before GALT1 upregulation, we did observe an increase in galactosylation, but also in sialylation, and this can be explained by the fact that ST6-GAL1 can only sialylate galactosylated glycans, so this may be a direct consequence of the increase in galactosylated glycans that we see. And at this point, we were actually ready to manipulate those candidate genes from that gene network that I showed you at the beginning. And these are the 6 genes that we manipulated. So at this point, we screened through about 20 GWAS hits, and only for these 6 did we observe a change in the glycan phenotype. So I will just go one by one and explain you what we observed. RUNX3 is actually a transcription factor that can either activate or repress gene expression regarding on the biological context. And upregulation of RUNX3 actually resulted in a decrease of galactosylated IgG glycans. This gene was a GWAS hit for galactosylation in one GWAS, but in another one it was associated with MGA3, so with bisecting GLCNAC. But we could not verify his role in bisection, only for galactosylation. Another gene that we targeted, and Azra already mentioned it, it's SPPL3. And SPPL3 downregulation resulted in the increase of both galactosylated and sialylated IgG glycans. And SPPL3 is a sheddase, and it can cleave GTs from the Golgi membranes, and it does that for MGAT5. And one study also showed that it does the same thing for B4GALT1. So what we observed in regards to galactosylation was pretty expected. But we see here also an increase in sialylation, and this locus was actually a GWAS hit for IgG sialylation. But again, this could be only a consequence of an increased galactosylation. Another gene is this HIV type 1 enhancer binding protein, and I said here it's associated with IgG degalactosylation. So what happened here is we could not upregulate nor downregulate this gene, but every time we did this experiment, every time we targeted the promoter of this gene, we observed a consistent increase of G2 glycans. So we are currently mapping the transcriptome to see if we inadvertently maybe affected some other locus that is actually implicated in IgG degalactosylation. Then we have MAMBA. This is mannozidase beta that works in lysosomes, and it cleaves many of the residues from N-link glycans. And its upregulation resulted in a decrease of G1 glycans, so monogalactosylated IgG glycans. And an interesting thing about MAMBA is that mutations in this gene are actually associated with cardiovascular diseases and hypertension, and G1 glycans turned out to be the best predictors of cardiovascular events in women. So MAMBA is definitely somehow implicated in monogalactosylation of IgG. We just have to find out in which way. Then we upregulated this TNF receptor superfamily member 13B, and its upregulation resulted in an increase of G0 IgG glycans. This enzyme is implicated in antibody production and B-cell differentiation, so it's feasible that maybe it can control the secretion of certain IgG glycoforms and in that way affect IgG glycosylation. And the last locus that we targeted is this EF1A1. It's an elongation factor, and its upregulation resulted in a decrease of G1 glycans. This is one of the most abundant proteins in cells, and it's implicated in a variety of signaling pathways. But one of the signaling pathways it controls is associated with interleukin-6 expression, and interleukin-6 is associated with IgG galactosylation, so that may be the way how this elongation factor affects IgG glycosylation. And while we were mapping all of these genes, another really interesting story emerged revolving around estrogen or estradiol. So we know that estrogen regulates IgG glycosylation, and we were wondering if we could map some of the downstream signaling mechanisms. So we probed our GWAS hits to see if some of them are regulated by estrogen, and it turned out that some of them indeed are. And most of them are associated with galactosylation or sialylation, and we know that estrogen affects galactosylation and sialylation. And then we prioritize 4 of the loci, and these are the results. So we manipulated every of those 4 loci, but only for RUNX3 and SPINK4 did we observe a change in the IgG glycan phenotype. And here you can see the results. So after upregulation of RUNX3 and SPINK4, we see a decrease of galactosylated IgG glycans. But an interesting thing is that upregulation of RUNX3 decreased both G1 and G2 glycans, but upregulation of SPINK4 decreased only G1 glycans, pointing to 2 regulatory mechanisms for IgG mono and degalactosylation. And this is something that we observe in population studies also. So for an example, in aging, degalactosylation decreases significantly while monogalactosylation stay fixed. So this is something that we are looking up right now. While we were doing all the experiments in those freestyle cells, we observed that glycome composition from in vitro secreted IgG changes with time, and we wanted to dig a bit deeper, so we did a time course experiment where we measured IgG glycans for approximately 100 days every 10 days, and here is what we've got. So it turned out that galactosylation was extremely variable, even though we tried to control for all the environmental factors while we were doing this study. All of the other traits turned out to be pretty stable. We did We did find some significant variation in fucosylation, but galactosylation was by far the most variable. And this is almost a cautionary tale because these cells are sometimes used to produce IgG with a defined glycan pattern. And here we see that galactosylation changes a lot even though we did nothing to the cells. But we were interested in another aspect here. We replicated this experiment, and it turned out that IgG galactosylation changes in the same way every time we did this experiment, which points to a non-random mechanism of change. And we are currently mapping the transcriptome to see if we could maybe find some regulators of this pathway that is responsible for the changes that we see. And to elucidate more mechanisms that revolve around IgG glycosylation, we are upgrading our system in a few ways. One of the things that we are doing, we are trying to look at regulation of Fab glycosylation. So everything we did that I talked about earlier is connected to Fc glycosylation, and now we obtain new vectors that contain also the Fab glycosylation site. And you can see here the profile in our freestyle cells. This is the Fc profile, and this is the Fab IgG glycan profile. And here are some annotated peaks. There are some crazy structures because, of course, these are freestyle cells. These are not really a model of B cells. So we are also trying to translate this technology to a better B cell model. One of them are these LCL cell lines, so lymphoblastoid cell lines, and we found a clone that secretes a lot of IgG, which is imperative for successful glycan analysis. And here you can see how that IgG profile looks like in this LCL clone. It differs a bit. It's represented in black here, and in blue is classical human glycan profile from IgG. And you can see there is a lot of this bisected core fucosylated structure, which is not that overrepresented in human glycome. We are in parallel doing the same thing. We are translating the technology to Ramos cell line. The difference is that LCLs endogenously secrete IgG, Ramos cells do not. And we are hoping, what we can do here is to actually define the sequence of IgG and maybe study different Fab glycosylation sites, how are they regulated, and so on. Another thing that we are doing, we are expanding our modular toolbox. So we started with just a few promoters, few fluorescent markers. We can now use many of them. We can also use almost an infinite number of guide RNAs, which is of course not true, but maybe around 15 or 20. We can target many genes at once, and we can target wide genomic regions with this bigger number of guide RNAs. And the most fun part for me is that we really expanded the effector domains that we can use. Right now, we started with these direct transcriptional regulators and DNA methyltransferase and demethylase, and now we have an array of different precise epigenome editors, ranging from histone lysine methyltransferases, histone lysine demethyltransferases, acetyltransferases, histone variants. So we can now actually deepen our research. We can now, for an example, look for those EVA studies and target the epigenome to see what we'll find out. And this is more or less it from me. Thank you.